Coastal zones are among the most productive environments on Earth. With the increased threats to marine biodiversity, there is a pressing need for more observations that can only be obtained from satellites. Yet coastal areas are also optically complex, which presents a significant challenge to the use of Earth observation missions. The HyperBOOST project was established to address this gap. Funded by the European Space Agency and operating within the framework of the EMBL TREC expedition, the primary objective was to collect high-quality measurements to validate the performance of current and prepare for future satellite missions, including the Sentinel-3 Ultra and upcoming Hyperspectral Chime mission.
Thanks to the collaboration between the European Space Agency and the European Commission projects, a unique dataset has been delivered. The institutional mechanisms put in place through the ESA and EC agreements have demonstrated in HyperBOOST and shown to be productive so that they can be extended into the future. Our methodology integrated continuous automated measurements with discrete station-based sampling across 120 coastal sites around the European coast, including Baltic, Atlantic and Mediterranean seas during 2023 and 2024. Utilizing the schooner Tara as a mobile research platform, we deployed a sophisticated suite of hyperspectral instruments continuously, sampling in the coastal waters. This included for the first time above-water radiometry from remote sensing reflectance and inline in-water intake for inherent optical properties already tested in previous Tara expeditions. But crucially, these optical data were paired with rigorous biogeochemical analysis, including pigment quantification and absorption measurements of phytoplankton. This collocated approach allowed for a direct match-up analysis between the in-situ reality and satellite-derived products, ensuring the highest standards of radiometric and biological product validation. And this was made possible thanks to an experienced team of experts, specialists in marine optics and remote sensing, from the HyperBOOST projects working in collaboration with biologists from EMBL and the Tara Ocean Foundation. This expertise, coupled with the technology and instrumentation on board of the Tara, make this first-of-its-kind sampling expedition possible and help us achieve a gold standard dataset for a variety of future science applications.
So such a big, large-scale database related to the wild European coastal waters was actually generated 25 years ago by the lab where I come from, Laboratoire de Sonography de Villefranche. It was done with all instruments, multispectral instruments, optical instruments notably. So the idea was to make such similar database, but using new technology, including hyperspectral optical measurements, to revisit in terms of hyperspectral, the optical properties and the biogeochemical properties of European coastal waters, so that we can develop new algorithms for the new hyperspectral satellite sensors observing the ocean.
So conventionally, satellite sensors have been multispectral, where they measure at a discrete number of spectral bands. Something like 8 to 10 in the visible spectrum is pretty typical. But as of now, that the NASA PACE satellite is launched, and there'll soon be the ESA CHIME satellite that will launch, which will measure information continuously across the visible spectrum. And this data set, which is publicly available for the whole community to use, is particularly well suited for developing mathematical relationships between the water constituents through to the inherent optical properties, through to the reflectance, through to the signal that we observe in space.
We use historical ocean color imagery and the satellite imagery of the couple of days before Tara was at sea in a given location to help the captain and the scientific people on board to decide exactly what was the best place where to stop to collect data, to make sure that whether it was the interest to collect turbid plume or whether the interest was to collect water in an offshore water not affected by the coastal influence, we could place the boat in the best place to achieve the research voyage mission.
The novel instrumentation we installed on board enabled us to take a large amount of continuous measurements throughout the expedition. One of the instruments crucial for the project was the SORAD, an automated continuous sampling system which recorded over 7,000 individual records. This was combined with systems measuring absorption and backscattering from seawater taken continuously from the ship's water intake.
The SORAD system, SORAD is an acronym for Solar Radiometry Tracking Platform, and the SORAD is an example of an optical system which measures the colour of the water above the water surface. It consists of 3 separate sensors, one of which points to the sky, one of which points to the water, and one of which measures the downwelling light. And from these 3 separate measurements, we derive what's termed remote sensing reflectance. And this is our measure of water colour. And from that, we can then work out the constituents of various particles and optically active traces that are in the water.
So I think that one really strength point of this dataset is that is very robust, because also the data quality of the replicates is amazing. There is nothing, I never saw something similar. So we cover areas where we don't have any data, and also we have all the areas covered with the same protocols, with the same approach. And all the samples were analyzed in the biophysics laboratories by the same people. This guarantee the total intercomparability of the data. So the dataset is very robust.
Thanks to HyperBOOST dataset, we are able to map regional differences in specific optical properties around the European coastal seas and to investigate advanced products from satellite related to phytoplankton diversity and carbon content. The data has also been instrumental to produce simulated data for the future ESA mission CHIME that will allow the community to have useful algorithms in place even before the satellite is launched. The impact of HyperBOOST extends beyond data validation. It represents a future bridge between the molecular biology of EMBL and the planetary scale observation of ESA. The results enhance our ability to monitor harmful algal blooms, track land to ocean carbon fluxes, and safeguarding marine biodiversity, therefore aligning with the European Space Agency New Science Strategy, published in 2024.
All HyperBOOST datasets are being integrated into global repositories to ensure long-term utility for the scientific community. As we look towards the launch of CHIME, the lessons learnt here will ensure that the European Space Agency remains at the forefront of research for improving and monitoring our understanding of the ocean.